Catalysts are substances that increase the rate of a chemical reaction without being consumed. They lower the activation energy by providing an alternate pathway for the reaction. Here we have an uncatalyzed reaction and a catalyzed reaction that takes place in 2 steps. The sum of the activation energy needed for each step in the catalyzed reaction is less than the energy needed in the uncatalyzed reaction, as seen in this diagram. Therefore, the rate is higher for the catalyzed reaction. Enzymes are biological catalysts that control the rates of chemical reactions within living cells, helping to sustain life. They typically speed up the reaction rate by 10 to the 8th to 10 to the 20th times. Cells contain thousands of different enzymes. Enzymes are proteins that function according to the sequence and types of amino acids in their chains. Lactase is an example of a biological catalyst found in the body. The lactase enzyme helps to break down and digest the sugar lactose. If the body doesn't produce enough lactase enzymes, lactose intolerance occurs. Each enzyme has a specific job and will only react with a certain substance. On their surface, they each have an empty pocket-like shape called the active site. The active site is where reactions with the enzyme occur. Enzymes react with molecules called substrates, which are typically smaller. The substrate fits into the pocket of the enzyme, and when they effectively collide, they become attached through intermolecular forces. The chemical change begins, and then product is formed, which is released by the enzyme. There are 2 different models of the process that occurs between the enzyme and substrate. One is the lock and key model. In this model, the fixed shape of the active site matches perfectly with the shape of the substrate, and the substrate fits right in like a key in a lock. The process that takes place according to the induced fit model occurs more often. In this model, the enzyme changes shape at the active site when it has come into contact with the substrate. Different factors can cause enzymes to denature. When enzymes denature, the shape of their active site changes, and they can no longer react with the substrate. These factors include temperature and pH. Enzymes are most active at certain temperatures. The optimal temperature is 98.6 degrees Fahrenheit, or body temperature. If the temperature is too high, the enzymes will stop working. Enzymes have various optimal pH values based on their structure and where they are located in the body. Changes in pH for any enzyme, however, can change its shape. Enzyme inhibitors can slow or stop an enzyme from catalyzing. The 3 most common forms of enzyme inhibition are competitive, non-competitive, and substrate inhibition. In competitive inhibition, the substrate competes with other substances for a spot in the enzyme's active site. The other substance is able to fit in the active site and block the substrate from being catalyzed. In non-competitive inhibition, a substance is added to the enzyme, which changes the enzyme's shape. It does not bond in the active site, but to another location, which then alters the shape of the active site and prevents substrates from being able to react. Substrate inhibition occurs when there is an oversupply of substrate molecules. There are so many of them that they block the entrance to the active sites and prohibit reactions from occurring.